What's up, guys? It's Dr. Murphy, Murphy Medical Associates, and our team at coronatestct.com. You are watching Murphy's Medical Minute. Thanks for joining. Today I'm going to talk about a couple of things. We're going to talk about the best vaccine, in my humble opinion, for the future. We're going to talk about the best vaccine for the future. We'll talk about the other vaccines as they roll out, but most importantly, as I covered before, the Johnson & Johnson vaccine. The Johnson & Johnson vaccine has been tested against the South African variant. It's been tested against the Brazilian P1 variant. It's been tested against the UK variant. In addition, today we're going to talk a little bit about the new variants that are out there. The new variants that are out there, predominantly not to be outdone by Los Angeles, but New York, you got yourself a new variant. You're watching Murphy's Medical Minute. Guys, thanks for joining. So one of the big things that we really need to think about is getting the right vaccine for the right person at the right time. It's just like any other sort of medical treatment. I think probably one of our biggest issues with it, though, and I'll tell you right now, is that we're getting new data all the time. We're getting new data all the time. And as we review the vaccines to find out which is best and which is worse, we have to match the vaccine up against the virus. Match the vaccine up against the novel SARS CoV2 coronavirus. We also have to think about the issues, the implications of people with immunosuppression. One of the problems with people who have immunosuppression is that they actually, when they get infected, they serve as reservoirs, harbors that allow the virus to mutate. So why aren't we vaccinating all the immunosuppressed first, and why aren't we testing vaccines solely on the immunosuppressed? Well, we are. We're testing vaccination against the virus for people who have HIV, and we've seen that. And in fact, the J&J vaccine is one of the leaders with that. The reason why we have to look at these vaccines is we are slowly starting to see vaccine escape. So what's vaccine escape? Well, when we have a mutation, right, in the receptor binding domain, the receptor binding domain, that's the hand, right, the pink stuff, the hand, or the N-terminal domain, we can see some vaccine escape. Vaccine escape matters as we get more variants. Maybe part 2 of vaccine escape, as you know, I talked a little bit about how the AstraZeneca vaccine had to be aborted for South African variant. That means they stopped the trial for the South African variant. Now, one of the other things that we've noticed here with all of this is some preliminary laboratory data on lack of sera induction and sera neutralization. What the heck does that mean? All right, so let me explain to you here, as you can see in this set of slides. The Pfizer vaccine induces an antibody response. That antibody response, right, you can see that antibody response right over there, right over there. That antibody response should neutralize a variant, And as you can see here, we have the list of the United Kingdom isolate, B117. We have the Denmark variant, B11298. We have what was a very prominent Los Angeles variant, B1429. The Manaus variant, P1. And the additional Brazilian isolate, P2. And then we have the South African variants. Predominantly B1351, V1, V2, and V3. We essentially found that after one dose of Pfizer vaccination, it does nothing for neutralization of the South African variant. We also found out that after two doses of the Pfizer vaccine, it does nothing against the South African variant, and it isn't terribly effective against the Brazilian variants. So the question you're going to ask yourself is the following. What do the Brazilian variant and the South African variant have in common? What do they have in common? Well, one of the things they have in common is the E484K mutation. The E484K mutation. Eek is the good and easy name to remember of it, because you should be eeked out about this. This is in the receptor binding domain. When we have the receptor binding domain mutated, we know that we can get escape from neutralizing antibodies, escape from sera. And in the case of the AstraZeneca vaccine, what we saw was very significant. What we saw was very, very significant. And I'll tell you right now, what we saw was nearly complete evasion of effect. Nearly complete evasion of effect. Evasion of neutralizing antibody. Right, we saw that. Remember, I talked about this before. Right, I talked about this before. We looked at vaccine escape. We looked at vaccine escape from the South African variant. We had to really be honest with ourselves and recognize that the B1351 variant was not going to be prevented from infecting humans with the AstraZeneca vaccine. However, we found that the Novavax vaccine and the J&J vaccine did have effect against the South African variant. The Novavax and the J&J vaccine, the Novavax and the J&J vaccine, remember the J&J vaccine is a DNA-based vaccine, and the Novavax vaccine is a protein-based vaccine. The AstraZeneca vaccine is a DNA vector-based vaccine as well, with the adenovirus. So these are not your mRNA vaccines. These are not your mRNA vaccines. The Novavax is a protein-based vaccine. The J&J and the AstraZeneca are adenoviral vector vaccines. The J&J vaccine had effect against the South African variant. So did the Novavax vaccine. Now let's review the mRNA vaccine data we have with Pfizer. The preliminary preprint data that came out shows that after two doses of the Pfizer vaccine, you did not achieve sera, blood, to produce antibodies. These antibodies did not neutralize the South African variant. So you're going to ask yourself, well, why the heck does this matter? Why does this matter? Well, I'm going to share with you right now why it matters. At least in the Northeast, where we are at coronatestct.com, coronatestnyc.com, where we're testing for novel coronavirus. We're also doing genomic surveillance in partnership with Wadsworth, and in partnership, that's New York State's lab, and in partnership with Yale and the School of Public Health. So the New York Times was lambasted even by me. Well, go figure, right? So the New York Times was lambasted by myself. Because I said, and I've always said, with the variants, just like when you go to a code for a patient, you need to check your own pulse first, right? We check our own pulse first before we check the patient, so we know our head is in the game. The rush to report on preprint is there. It's there. Take a look. Who's the first guy to report on the laboratory finding that the South African variant probably is not going to be effectively neutralized by Pfizer vaccine? Hi. It was me. The day it went on medRxiv. So the New York Times, not to be outdone, reported first on this variant up here, B1526. B1526. Wait a second. Now you're talking about all these crazy things. All right, so let's talk about New York, New York. Right? Not to be outdone by Los Angeles, New York has come up with their own special variant. Congratulations, New York. You ruined coronavirus. I say this in a little bit of jest, but I have to be deadly serious about it. B1526 has E484K mutation. It has the E484K mutation. Has the E484K mutation. And it is spreading in New York City. Everyone thought that the British variant would be the variant that would spread like wildfire all over the place in the United States. Is typical in American fashion, we have rejected the British. Hooray! Right? We have rejected the British, the United Kingdom variant, the isolate, and instead we have ended up with our own special homebrew. Our own special homebrew. One of those is the Californian isolate, the Californian variant, and more concerning is right there. The New York variant B1526, originally reported in the New York Times. Before anyone had a chance to review the data, they spit it out. Can't deposit excuses, and I'll take the hit myself. I reported before anyone else also had to review this data. Luckily, I'm not a scientific journalist. I'm only a geneticist, a doctor, a researcher, an educator. So luckily, I had a little bit of wherewithal to be able to review the preliminary data on the ineffectiveness of the Pfizer vaccine. The ineffectiveness of the Pfizer vaccine. And so I felt comfortable going here on this channel, my channel, Murphy's Medical Minute, to review that. I really have been of the point where I say CTFO. When it comes to variants, CTFO. We have to study and we have to review the variants. I am now coming on Murphy's Medical Minute to say the following. The variant B1526, in my mind, is a variant of concern. It's a variant of concern. And we need to be very serious about this when we go through and review vaccination. When we go through and review vaccination. Let me tell you why. You ready for why? The New York City variant has some of the mutations the South African variant has, including a couple of homebrew special mutations on its own, which are very concerning. These are very concerning. We have mutations in the receptor binding domain. We got a lot of mutations. I'm going to go through this briefly with you just to know that we have mutations where we could escape antibody neutralization. We have mutations that can enhance being able to infect a cell. We have the South African variant mutations. And this special homebrew in New York, right? You guys watched the intro with me in the Yankees ring. God, we need another championship. If you're listening, Booney, if you're listening to me, Booney, it's time to buckle down with those boys. Time to buckle down with those boys. I know my good friend, Michael K, will say, hey, there's no excuses. There's no excuses. And in this case, you can't deposit excuses. So we've got to get a new championship. Have to. And we've got to get fans back in Yankee Stadium. But with this new variant, I have deep concern. Let me tell you why. This new variant here has mutations in all the wrong places. Mutations in all the wrong places. And what we're really seeing with this is a couple of things. But first, I want to answer a couple of questions. Someone on Periscope just said to me, they'll have to beat the Cards. They'll beat the Cards. I'm not worried about the Yankees this year. They'll beat the Cards. So, in all seriousness, we have to ask ourselves, well, where did this B1526 variant come from? And in a recently published preprint medRxiv review, we find our good friends at Columbia School of Physicians and Surgeons and Columbia University, where they dig through their biobank from November to February 15th. So they dug through all of the samples, just like us at coronatestct.com. We freeze our positives. We biobank our positives, and we send our positives to our laboratory partners that do genomic sequencing with us. Most notably, the Yale School of Public Health and Yale University. Thank you very much. Not to say that Yale is in a bloodbath duel with Columbia, but they're different institutions. Different institutions. So we partner with Wadsworth in New York State for our New York State samples, and we partner with Yale School of Public Health, the Grubau Lab, and Yale University for our Connecticut samples. And we've been very happy with these partnerships because we are leading the edge in both states with genomic surveillance. We are the tip of the spear. Why? Because of Yale University and Wadsworth. So I want to thank both of them, Yale University and Wadsworth, for helping us so much with our genomic surveillance. They're just doing a great job. So where did they find these New York variants? Now announcing the New York variants. And in this case, B1526. This is the variant I believe should be a variant of concern as we review it, merely because it has the same mutation, E484K, that the South African variant has, right? That the South African variant has, B1351, and also that the P1 Manaus and P2 variants have. This mutation in the receptor binding domain is causing some fits, and so we're concerned a little bit about it. In addition, we're also concerned about the N-terminal domain mutations that we see. I don't know if you guys can take a look here. Right, you take a look up at the top. Up at the top. This is the biobank registry of samples from Columbia. Take a look. Take a look at the magenta bar. Take a look at the magenta bar. You see the magenta bar up here? The magenta set of samples? It's like 25% of all samples that are in the Columbia biobank. 25%. 1 in 4 samples in the Columbia biobank are of this New York variant that has E484K. And I don't know if you can take a look, but look at the growth. Look at the growth between January and February of this variant. Look at the growth between January and February of this variant. So the Columbia Biobank is a bunch of diverse neighborhoods in New York City. A bunch of neighborhoods in New York City. If you were someone who got the Pfizer vaccines, I think I demonstrated some concern that we have with Pfizer being able to fight off the E484K variant mutation. I think I might consider another vaccination. It's going to sound crazy. But if you got the Pfizer vaccination and are in the New York City area, I'd either consider a booster when that ever comes out, or get the J&J vaccine. Maybe get the J&J vaccine. So the patients they identified with the New York City variant, B1526, from the Columbia Biobank were older patients, 50 plus, and they were more likely to be hospitalized. These patients were more likely to be hospitalized. The New York variant is in the Northeast. B1526 is located in other locations in the Northeast, but a lot of the samples in New York City in the Columbia Biobank are B1526. So I believe that that variant will rip through New York City. I do. I do. And I think a lot of things out of New York City come from Washington Heights. And in this case, we have to be serious about our immunization strategy, especially in Washington Heights. If I were the New York City Citywide Immunization Registry and the team at New York City hospitals, I would be pumping J&J into Washington Heights. I would be pumping J&J into Washington Heights. Now, the reason I'm concerned is the following. P1 and P2 in Brazil, the Manaus isolate in Brazil and P2 have reinfections. They have reinfections. That's confirmed. We have been not reporting a lot on the Brazilian variant because I have been focused on the South African variant because that's where the data is. Well, the data is now building in Brazil. The Brazilian team is doing a fantastic job with genomic surveillance. Here's the problem. In the United States, we are not doing a great job with genomic surveillance. I think one of the big problems that we have with genomic surveillance is, put simply, that these genomes are not getting reimbursed. Now, President Biden has committed a bunch of money to genetic testing, a bunch of money to testing for SARS CoV2. Where will that money go? Where will that money funnel? And one of the big problems I find with governmental financing is it's like the dean's tax at any academic university. The school pulls, it grabs like the receptor binding domain of SARS CoV2 and it says, come here, let me take my share. And so it takes its piece off the top. Now, as you start with the governmental money, it trickles down, everyone taking a piece out of it until it gets to the source. This is true with healthcare today, but it is especially true with academia as well. Everyone's pulling a piece of that money before it ever gets to the researcher. The same thing is true with physicians. I can tell you personally from experience, yeah, I'm totally going to publish, sorry about that. Someone asked me to publish the archive. All of the notes, all of the studies, all of the links will be published down in my YouTube. So you can look at down here at the links. I'll put some links in my comment section, YouTube. You can follow up just with some more information so you can see what's going on. But I'm going to tell you today, the state of financing of research and the state of financing of coronavirus testing is a mess in the United States. It's a mess in the United States. We had Medicare have to come out and slap the insurance companies on the wrist and tell them that they have to pay for testing. What? Yeah. Insurance companies all over the place are stiffing doctors everywhere. They're stiffing laboratories, they're stiffing doctors. The same thing is true with the research dollars committed to universities. The university takes its money out first before it ever gets to the researcher. So the laboratory is losing a full third of its resource dollars for the institution. Boink! Pulling it right out. The dean's tax. Have a nice day. It's just the way that it is in these operations. All of this has to be fixed. Otherwise, we're not going to get to full genomic surveillance, which is really what we need here. We need a better genomic surveillance strategy. We need it at a state level. We need it at a national level. Why? Because of the variants, right? Look at these guys. Because of the variants. We now, I believe, have a new variant of concern in the New York City variant. B1526, which I would be happy calling New York's finest, right? We call it New York's finest. Now, the important part that you need to remember about B1526 is it has mutations similar to the Brazilian and the South African variants. And I don't like to conjecture. I don't like to I try not to do that. And I don't want to call the New York City variant the same as the Brazilian and the South African mashed up, because it's not. It's not the same. It's not a mashup of the two. It's not a collab. It's not a YouTube collab between B1351 and P1. It's not a collab. It is its own variant. It's its own variant. And you can see here as you drop down from it, it is closest to B1351, the South African variant. And so when I look at that data, I have to try and pick which is the best vaccine. I showed some data before that are pretty consistent. The Novavax vaccine and the J&J vaccine are our best options against the South African variant. They are our best options against the South African variant. Based on the data, I believe that the J&J vaccine that we have right now is our best option against the New York City variant. It's what the data indicate. We still have to study it, but if you are a scientist, posing a hypothesis, I would hypothesize that the effect with the J&J vaccine would be similar in B1526, the New York City variant, with B1351, the South African variant. And you can take a look here, right? So take a look. Let's see. Oh, right there. Right here. We're going to go right here. Right down there. You see that right down there? That's a lineage. It's a phylogenetic tree of relatedness. B1351 is similar, not the same, is similar to B1526. South Africa and New York City have a similar makeup. They are phylogenetically similar. Therefore, I would believe that J&J and Novavax are going to win the day with the B1526 variant. And that's my take. I'm sticking with it, guys. I'm absolutely sticking with it. It makes no sense here to mince words as I wrap into my 30 minutes of Murphy's Medical Minute. It's kind of a bait and switch, right? It's a bait and switch of this live stream. Today, I actually want to talk about different types of masks and the best masks for you. And I'm here to tell you right now that the best masks are the N95s. We need to be producing N95s on a large scale. They need to come into New York City, where we have a very scary variant called B1526. Let me rephrase the word. I kind of want to be almost like dead inside and just lead with the data. The B1526 variant has been found predominantly in a couple of neighborhoods around Columbia University in Washington Heights. It's also been scattered around New York City in a couple different places. Most of the B1526 variants were found in older and hospitalized patients with the Columbia Biobank. The number of samples that are increasing in the Columbia Biobank are significant. In January, it went for a very small amount. Till now, it's 1 in 4. 1 in 4. I believe that if we don't get a hold of this in New York City, it will grow. Yep, B1526 will grow. B1526 has the E484K mutation, similar to the South African variant and similar to the Brazilian P1 and P2 variants. The Brazilian variants are causing reinfection. The Brazilian variants are being researched right now. We're going to have more data on them. But one thing we can be certain about is that the Brazilian variants cause reinfection. The South African variant has more data. The South African variant is more similar to B1526 phylogenetically. The South African variant is not effectively attacked and neutralized by the AstraZeneca vaccine. Therefore, the Oxford AstraZeneca vaccine trial has stopped in South Africa or places where B1351 are. South African variant is here in the United States as well. We know that. It's established fact. The New York City variant is here as well in the Northeast, almost exclusively in the Northeast, where we are at coronatestct.com. My team is actively doing genomic surveillance. We are actively struggling to get vaccines. We have applied and have been waiting for weeks to get our vaccinations. I've got freezer data. We can hold the Pfizer vaccine. We can hold the Moderna vaccine. We can hold the J&J vaccine. We're ready to go. We're chomping at the bit. And in fact, we are dying to get our hands on the J&J vaccine. I'm going to take the J&J vaccine because it has data against the South African variant. Has data against the Brazilian variant as well. So I will choose the J&J vaccine for me. The best vaccine for you is the one you can get in your arm. The second best vaccine for you is the one that has coverage of the coronavirus variants that are where you live. If you're in New York City, that's J&J for me. If you're in New York City, J&J is the king. J&J is the king here. Novavax will be the queen. If you've gotten the Pfizer vaccines and you're in Washington Heights, you should seriously consider getting another shot with the J&J vaccine. All right. So now we're at 31 minutes. This is Dr. Murphy, Murphy Medical Associates, and our team at coronatestct.com. Thank you so much for watching Murphy's Medical Minute. Thank you so much for watching Murphy's Medical Minute. I'm very excited about being able to have this YouTube channel. I want to thank Periscope for shutting down so I could create Murphy's Medical Minute. To all my Periscope peeps, thank you very much. Right, they're all asking me for J&J vaccine, which I think is a smart move right now, especially if you're in New York City, which we are. I want to thank you again. Get outside. It's sunny out. It's beautiful. Go for a walk. It's cold in the Northeast, so bundle up. Take your vitamin D. There's no excuse to be vitamin D deficient. No excuse. You need to take care of your body. Exercise, eat right, get good sleep, and get good sun. Our bodies are made to be well. Thank you again for watching Murphy's Medical Minute. And to my Disney peeps, stand clear of the closing doors, please. With